Neural oscillation is rhythmic or repetitive neural activity in the central nervous system. Neural tissue can generate oscillatory activity in many ways, driven either by mechanisms within individual neurons or by interactions between neurons. In individual neurons, oscillations can appear either as oscillations in membrane potential or as rhythmic patterns of action potentials, which then produce oscillatory activation of post-synaptic neurons. At the level of neural ensembles, synchronized activity of large numbers of neurons can give rise to macroscopic oscillations, which can be observed in the electroencephalogram. Oscillatory activity in groups of neurons generally arises from feedback connections between the neurons that result in the synchronization of their firing patterns. The interaction between neurons can give rise to oscillations at a different frequency than the firing frequency of individual neurons. A well-known example of macroscopic neural oscillations is alpha activity. Neural oscillations were observed by researchers as early as 1924. More than 50 years later, intrinsic oscillatory behavior was encountered in vertebrate neurons, but its functional role is still not fully understood. The possible roles of neural oscillations include feature binding, information transfer mechanisms and the generation of rhythmic motor output. Over the last decades, more insight has been gained, especially with advances in brain imaging. A major area of research in neuroscience involves determining how oscillations are generated and what their roles are. Oscillatory activity in the brain is widely observed at different levels of observation and is thought to play a key role in processing neural information. Numerous experimental studies support a functional role of neural oscillations. A unified interpretation, however, is still lacking. Overview: neural oscillations are observed throughout the central nervous system and at all levels, for example, spike trains, local field potentials and large-scale oscillations which can be measured by electroencephalography. In general, oscillations can be characterized by their frequency, amplitude and phase. These signal properties can be extracted from neural recordings using time-frequency analysis. In large-scale oscillations, amplitude changes are considered to result from changes in synchronization within a neural ensemble, also referred to as local synchronization. In addition to local synchronization, oscillatory activity of distant neural structures can synchronize. Neural oscillations and synchronization have been linked to many cognitive functions such as information transfer, perception, motor control and memory. Neural oscillations have been most widely studied in neural activity generated by large groups of neurons. Large-scale activity can be measured by techniques such as electroencephalography. In general, EEG signals have a broad spectral content similar to pink noise, but also reveal oscillatory activity in specific frequency bands. The first discovered and best-known frequency band is alpha activity that can be detected from the occipital lobe during relaxed wakefulness and increases when the eyes are closed. Other frequency bands are delta, theta, beta and gamma frequency band, where faster rhythms such as gamma activity have been linked to cognitive processing. Indeed, EEG signals change dramatically during sleep and show a transition from faster frequencies to increasingly slower frequencies such as alpha waves. In fact, different sleep stages are commonly characterized by their spectral content. Consequently, neural oscillations have been linked to cognitive states, such as awareness and consciousness. Although neural oscillations in human brain activity are mostly investigated using EEG recordings, they are also observed using more invasive recording techniques such as single-unit recordings. Neurons can generate rhythmic patterns of action potentials or spikes. Some types of neurons have the tendency to fire at particular frequencies, so-called resonators. Bursting is another form of rhythmic spiking. Spiking patterns are considered fundamental for information coding in the brain. Oscillatory activity can also be observed in the form of subthreshold membrane potential oscillations. If numerous neurons spike in synchrony, they can give rise to oscillations in local field potentials. Quantitative models can estimate the strength of neural oscillations in recorded data. Neural oscillations are commonly studied from a mathematical framework and belong to the field of neurodynamics, an area of research in the cognitive sciences that places a strong focus upon the dynamic character of neural activity in describing brain function. It considers the brain a dynamical system and uses differential equations to describe how neural activity evolves over time. In particular, it aims to relate dynamic patterns of brain activity to cognitive functions such as perception and memory. In very abstract form, neural oscillations can be analyzed analytically. When studied in a more physiologically realistic setting, oscillatory activity is generally studied using computer simulations of a computational model. The functions of neural oscillations are wide-ranging and vary for different types of oscillatory activity. Examples are the generation of rhythmic activity such as a heartbeat and the neural binding of sensory features in perception, such as the shape and color of an object. Neural oscillations also play an important role in many neurological disorders, such as excessive synchronization during seizure activity in epilepsy or tremor in patients with Parkinson's disease. Oscillatory activity can also be used to control external devices in brain-neurocomputer interfaces, in which subjects can control an external device by changing the amplitude of particular brain rhythmics. Physiology. Oscillatory activity is observed throughout the central nervous system at all levels of organization. 3 different levels have been widely recognized: the microscale, the mesoscale and the macroscale. Microscopic neurons generate action potentials resulting from changes in the electric membrane potential. Neurons can generate multiple action potentials in sequence, forming so-called spike trains. These spike trains are the basis for neural coding and information transfer in the brain. Spike trains can form all kinds of patterns, such as rhythmic spiking and bursting, and often display oscillatory activity. Oscillatory activity in single neurons can also be observed in subthreshold fluctuations in membrane potential. These rhythmic changes in membrane potential do not reach the critical threshold and therefore do not result in an action potential. They can result from post-synaptic potentials from synchronous inputs or from intrinsic properties of neurons. Neuronal spiking can be classified by their activity patterns. The excitability of neurons can be subdivided in class I and II. Class I neurons can generate action potentials with arbitrarily low frequency depending on the input strength, whereas class II neurons generate action potentials in a certain frequency band, which is relatively insensitive to changes in input strength. Class II neurons are also more prone to display subthreshold oscillations in membrane potential. Mesoscopic a group of neurons can also generate oscillatory activity. Through synaptic interactions, the firing patterns of different neurons may become synchronized and the rhythmic changes in electric potential caused by their action potentials will add up. That is, synchronized firing patterns result in synchronized input into other cortical areas, which gives rise to large amplitude oscillations of the local field potential. These large-scale oscillations can also be measured outside the scalp using electroencephalography and magnetoencephalography. The electric potentials generated by single neurons are far too small to be picked up outside the scalp, and EEG or MEG activity always reflects the summation of the synchronous activity of thousands or millions of neurons that have similar spatial orientation. Neurons in a neural ensemble rarely all fire at exactly the same moment, that is fully synchronized. Instead, the probability of firing is rhythmically modulated such that neurons are more likely to fire at the same time, which gives rise to oscillations in their mean activity. As such, the frequency of large-scale oscillations does not need to match the firing pattern of individual neurons. Isolated cortical neurons fire regularly under certain conditions, but in the intact brain cortical cells are bombarded by highly fluctuating synaptic inputs and typically fire seemingly at random. However, if the probability of a large group of neurons is rhythmically modulated at a common frequency, they will generate oscillations in the mean field. Neural ensembles can generate oscillatory activity endogenously through local interactions between excitatory and inhibitory neurons. In particular, Inhibitory interneurons play an important role in producing neural ensemble synchrony by generating a narrow window for effective excitation and rhythmically modulating the firing rate of excitatory neurons. Macroscopic neural oscillation can also arise from interactions between different brain areas. Time delays play an important role here. Because all brain areas are bidirectionally coupled, these connections between brain areas form feedback loops. Positive feedback loops tends to cause oscillatory activity, which frequency is inversely related to the delay time. An example of such a feedback loop is the connections between the thalamus and cortex. This thalamocortical network is able to generate oscillatory activity known as recurrent thalamo-cortical resonance. The thalamocortical network plays an important role in the generation of alpha activity. Mechanisms neuronal properties Scientists have identified some intrinsic neuronal properties that play an important role in generating membrane potential oscillations. In particular, voltage-gated ion channels are critical in the generation of action potentials. The dynamics of these ion channels have been captured in the well-established Hodgkin-Huxley model that describes how action potentials are initiated and propagated by means of a set of differential equations. Using bifurcation analysis, different oscillatory varieties of these neuronal models can be determined, allowing for the classification of types of neuronal responses. The oscillatory dynamics of neuronal spiking as identified in the Hodgkin-Huxley model closely agree with empirical findings. In addition to periodic spiking, subthreshold membrane potential oscillations, that is resonance behavior that does not result in action potentials, may also contribute to oscillatory activity by facilitating synchronous activity of neighboring neurons. Like pacemaker neurons in central pattern generators, subtypes of cortical cells fire bursts of spikes rhythmically at preferred frequencies. Bursting neurons have the potential to serve as pacemakers for synchronous network oscillations, and bursts of spikes may underlie or enhance neuronal resonance. Network properties Apart from intrinsic properties of neurons, network properties are also an important source of oscillatory activity. Neurons communicate with one another via synapses and affect the timing of spike trains in the post-synaptic neurons. Depending on the properties of the connection, such as the coupling strength, time delay and whether coupling is excitatory or inhibitory, the spike trains of the interacting neurons may become synchronized. Neurons are locally connected, forming small clusters that are called neural ensembles. Certain network structures promote oscillatory activity at specific frequencies. For example, neuronal activity generated by two populations of interconnected inhibitory and excitatory cells can show spontaneous oscillations that are described by the Wilson-Cowan model. If a group of neurons engages in synchronized oscillatory activity, the neural ensemble can be mathematically represented as a single oscillator. Different neural ensembles are coupled through long-range connections and form a network of weakly coupled oscillators at the next spatial scale. Weakly coupled oscillators can generate a range of dynamics including oscillatory activity. Long-range connections between different brain structures, such as the thalamus and the cortex, involve time delays due to the finite conduction velocity of axons. Because most connections are reciprocal, they form feedback loops that support oscillatory activity. Oscillations recorded from multiple cortical areas can become synchronized and form a large-scale network, whose dynamics and functional connectivity can be studied by means of spectral analysis and Granger causality measures. Coherent activity of large-scale brain activity may form dynamic links between brain areas required for the integration of distributed information. Neuromodulation. In addition to fast direct synaptic interactions between neurons forming a network, oscillatory activity is modulated by neurotransmitters on a much slower time scale. That is, the concentration levels of certain neurotransmitters are known to regulate the amount of oscillatory activity. For instance, GABA concentration has been shown to be positively correlated with frequency of oscillations in induced stimuli. A number of nuclei in the brainstem have diffuse projections throughout the brain influencing concentration levels of neurotransmitters such as norepinephrine, acetylcholine and serotonin. These neurotransmitter systems affect the physiological state, for example, wakefulness or arousal, and have a pronounced effect on amplitude of different brain waves, such as alpha activity. Mathematical description. Oscillations can often be described and analyzed using mathematics. Mathematicians have identified several dynamical mechanisms that generate rhythmicity. Among the most important are harmonic oscillators, limit cycle oscillators, and delayed feedback oscillators. Harmonic oscillations appear very frequently in natural examples are sound waves, the motion of a pendulum, and vibrations of every sort. They generally arise when a physical system is perturbed by a small degree from a minimum energy state, and are well understood mathematically. Noise-driven harmonic oscillators realistically simulate alpha rhythm in the waking EEG as well as slow waves and spindles in the sleep EEG. Successful EEG analysis algorithms were based on such models. Several other EEG components are better described by limit cycle or delayed feedback oscillations. Limit cycle oscillations arise from physical systems that show large deviations from equilibrium, whereas delayed feedback oscillations arise when components of a system affect each other after significant time delays. Limit cycle oscillations can be complex, but there are powerful mathematical tools for analyzing them. The mathematics of delayed feedback oscillations is primitive in comparison. Linear oscillators and limit cycle oscillators qualitatively differ in terms of how they respond to fluctuations in input. In a linear oscillator, the frequency is more or less constant but the amplitude can vary greatly. In a limit cycle oscillator, the amplitude tends to be more or less constant but the frequency can vary greatly. A heartbeat is an example of a limit cycle oscillation in that the frequency of beats varies widely, while each individual beat continues to pump about the same amount of blood. Computational models adopt a variety of abstractions in order to describe complex oscillatory dynamics observed in brain activity. Many models are used in the field, each defined at a different level of abstraction and trying to model different aspects of neural systems. They range from models of the short-term behavior of individual neurons, through models of how the dynamics of neural circuitry arise from interactions between individual neurons, to models of how behavior can arise from abstract neural modules that represent complete subsystems. Single neuron model equals. A model of a biological neuron is a mathematical description of the properties of nerve cells, or neurons, that is designed to accurately describe and predict its biological processes. The most successful and widely used model of neurons, the Hodgkin-Huxley model, is based on data from the squid giant axon. It is a set of nonlinear ordinary differential equations that approximates the electrical characteristics of a neuron, in particular the generation and propagation of action potentials. The model is very accurate and detailed, and Hodgkin and Huxley received the 1963 Nobel Prize in Physiology or Medicine for this work. The mathematics of the Hodgkin-Huxley model are quite complicated and several simplifications have been proposed, such as the Fitzhugh-Nagumo model and the Hind-Marshall-Rose model. Such models only capture the basic neuronal dynamics, such as rhythmic spiking and bursting, but are more computationally efficient. This allows the simulation of a large number of interconnected neurons that form a neural network. Spiking model equals. A neural network model describes a population of physically interconnected neurons or a group of disparate neurons whose inputs or signaling targets define a recognizable circuit. These models aim to describe how the dynamics of neural circuitry arise from interactions between individual neurons. Local interactions between neurons can result in the synchronization of spiking activity and form the basis of oscillatory activity. In particular, models of interacting pyramidal cells and inhibitory interneurons have been shown to generate brain rhythms such as gamma activity. Neural mass model equals. Neural field models are another important tool in studying neural oscillations and are a mathematical framework describing evolution of variables such as mean firing rate in space and time. In modeling the activity of large numbers of neurons, the central idea is to take the density of neurons to the continuum limit, resulting in spatially continuous neural networks. Instead of modeling individual neurons, this approach approximates a group of neurons by its average properties and interactions. It is based on the mean field approach, an area of statistical physics that deals with large-scale systems. Models based on these principles have been used to provide mathematical descriptions of neural oscillations and EEG rhythms. They have, for instance, been used to investigate visual hallucinations. Kuramoto model equals. The Kuramoto model of coupled phase oscillators is one of the most abstract and fundamental model used to investigate neural oscillations and synchronization. It captures the activity of a local system by its circular phase alone and hence ignores the amplitude of oscillations. Interactions amongst these oscillators are introduced by a simple algebraic form and collectively generate a dynamical pattern at the global scale. The Kuramoto model is widely used to study oscillatory brain activity and several extensions have been proposed that increase its neurobiological plausibility, for instance by incorporating topological properties of local cortical connectivity. In particular, it describes how the activity of a group of interactioning neurons can become synchronized and generate large-scale oscillations. Simulations using the Kuramoto model with realistic long-range cortical connectivity and time-delayed interactions reveal the emergence of slow-patterned fluctuations that reproduce resting-state bold functional maps, which can be measured using fMRI. Activity patterns, both single neurons and groups of neurons can generate oscillatory activity spontaneously. In addition, they may show oscillatory responses to perceptual input or motor output. Some types of neurons will fire rhythmically in the absence of any synaptic input. Likewise, brain-wide activity reveals oscillatory activity while subjects do not engage in any activity, so-called resting state activity. These ongoing rhythms can change in different ways in response to perceptual input or motor output. Oscillatory activity may respond by increases or decreases in frequency and amplitude or show a temporary interruption, which is referred to as phase resetting. In addition, external activity may not interact with ongoing activity at all, resulting in an additive response. Ongoing activity equals. Spontaneous activity is brain activity in the absence of an explicit task, such as sensory input or motor output, and hence also referred to as resting state activity. It is opposed to induced activity, that is brain activity that is induced by sensory stimuli or motor responses. The term ongoing brain activity is used in electroencephalography and magnetoencephalography for those signal components that are not associated with the processing of a stimulus or the occurrence of specific other events, such as moving a body part, that is events that do not form evoked potentials, evoked fields, or induced activity. Spontaneous activity is usually considered to be noise if one is interested in stimulus processing. However, spontaneous activity is considered to play a crucial role during brain development, such as in network formation and synaptogenesis. Spontaneous activity may be informative regarding the current mental state of the person and is often used in sleep research. Certain types of oscillatory activity, such as alpha waves, are part of spontaneous activity. Statistical analysis of power fluctuations of alpha activity reveals a bimodal distribution, that is a high and low amplitude mode, and hence shows that resting state activity does not just reflect a noise process. In case of fMRI, spontaneous fluctuations in the blood oxygen level dependent signal reveal correlation patterns that are linked to resting states networks, such as the default network. The temporal evolution of resting state networks is correlated with fluctuations of oscillatory EEG activity in different frequency bands. Ongoing brain activity may also have an important role in perception, as it may interact with activity related to incoming stimuli. Indeed, EEG studies suggest that visual perception is dependent on both the phase and amplitude of cortical oscillations. For instance, the amplitude and phase of alpha activity at the moment of visual stimulation predicts whether a weak stimulus will be perceived by the subject. Frequency response equals, in response to input, A neuron or neuronal ensemble may change the frequency at which it oscillates, thus changing the rate at which it spikes. Often, a neuron's firing rate depends on the summed activity it receives. Frequency changes are also commonly observed in central pattern generators and directly relate to the speed of motor activities, such as step frequency in walking. However, changes in relative oscillation frequency between different brain areas is not so common because the frequency of oscillatory activity is often related to the time delays between brain areas. Phase resetting Phase resetting occurs when input to a neuron or neuronal ensemble resets the phase of ongoing oscillations. It is very common in single neurons where spike timing is adjusted to neuronal input and may also occur in neuronal ensembles when the phases of their neurons are adjusted simultaneously. Phase resetting is fundamental for the synchronization of different neurons or different brain regions because the timing of spikes can become phase-locked to the activity of other neurons. Phase resetting also permits the study of evoked activity, a term used in electroencephalography and magnetoencephalography for responses in brain activity that are directly related to stimulus-related activity. Evoked potentials and event-related potentials are obtained from an electroencephalogram by stimulus-locked averaging, that is averaging different trials at fixed latencies around the presentation of a stimulus. As a consequence, those signal components that are the same in each single measurement are conserved and all others, that is ongoing or spontaneous activity, are averaged out. That is, event-related potentials only reflect oscillations in brain activity that are phase-locked to the stimulus or event. Evoked activity is often considered to be independent from ongoing brain activity, although this is an ongoing debate. Amplitude response next to evoked activity, neural activity related to stimulus processing may result in induced activity. Induced activity refers to modulation in ongoing brain activity induced by processing of stimuli or movement preparation. Hence, they reflect an indirect response in contrast to evoked responses. A well-studied type of induced activity is amplitude change in oscillatory activity. For instance, gamma activity often increases during increased mental activities such as during object representation. Because induced responses may have different phases across measurements and therefore would cancel out during averaging, they can only be obtained using time-frequency analysis. Induced activity generally reflects the activity of numerous neurons. Amplitude changes in oscillatory activity are thought to arise from the synchronization of neural activity, for instance by synchronization of spike timing or membrane potential fluctuations of individual neurons. Increases in oscillatory activity are therefore often referred to as event-related synchronization, while decreases are referred to as event-related desynchronization. Asymmetric amplitude modulation. It has recently been proposed that even if phases are not aligned across trials, induced activity may still cause event-related potentials because ongoing brain oscillations may not be symmetric and thus amplitude modulations may result in a baseline shift that does not average out. This model implies that slow event-related responses, such as asymmetric alpha activity, could result from asymmetric brain oscillation amplitude modulations, such as an asymmetry of the intracellular currents that propagate forward and backward down the dendrites. Under this assumption, asymmetries in the dendritic current would cause asymmetries in oscillatory activity measured by EEG and MEG, since dendritic currents in pyramidal cells are generally thought to generate EEG and MEG signals that can be measured at the scalp. Function. Neural synchronization can be modulated by task constraints, such as attention, and is thought to play a role in feature binding, neuronal communication, and motor coordination. Neuronal oscillations became a hot topic in neuroscience in the 1990s when the studies of the visual system of the brain by Gray, Singer and others appeared to support the neural binding hypothesis. According to this idea, synchronous oscillations in neuronal ensembles bind neurons representing different features of an object. For example, when a person looks at a tree, visual cortex neurons representing the tree trunk and those representing the branches of the same tree would oscillate in synchrony to form a single representation of the tree. This phenomenon is best seen in local field potentials which reflect the synchronous activity of local groups of neurons, but has also been shown in EEG and MEG recordings, providing increasing evidence for a close relation between synchronous oscillatory activity and a variety of cognitive functions such as perceptual grouping. Pacemaker equals. Cells in the sinoatrial node, located in the right atrium of the heart, spontaneously depolarize approximately 100 times per minute. Although all of the heart cells have the ability to generate action potentials that trigger cardiac contraction, the sinoatrial node normally initiates it, simply because it generates impulses slightly faster than the other areas. Hence, these cells generate the normal sinus rhythm and are called pacemaker cells as they directly control the heart rate. In the absence of extrinsic neural and hormonal control, cells in the SA node will rhythmically discharge. The sinoatrial node is richly innervated by the autonomic nervous system, which up or down regulates the spontaneous firing frequency of the pacemaker cells. Central pattern generator equals. Synchronized firing of neurons also forms the basis of periodic motor commands for rhythmic movements. These rhythmic outputs are produced by a group of interacting neurons that form a network, called a central pattern generator. Central pattern generators are neuronal circuits that, when activated, can produce rhythmic motor patterns in the absence of sensory or descending inputs that carry specific timing information. Examples are walking, breathing, and swimming. Most evidence for central pattern generators comes from lower animals, such as the lamprey, but there is also evidence for spinal central pattern generators in humans. Information processing equals. Neuronal spiking is generally considered the basis for information transfer in the brain. For such a transfer, information needs to be coded in a spiking pattern. Different types of coding schemes have been proposed, such as rate coding and temporal coding. Perception equals. Synchronization of neuronal firing may serve as a means to group spatially segregated neurons that respond to the same stimulus in order to bind these responses for further joint processing, that is to exploit temporal synchrony to encode relations. Purely theoretical formulations of the binding by synchrony hypothesis were proposed first, but subsequently extensive experimental evidence has been reported supporting the potential role of synchrony as a relational code. The functional role of synchronized oscillatory activity in the brain was mainly established in experiments performed on awake kittens with multiple electrodes implanted in the visual cortex. These experiments showed that groups of spatially segregated neurons engage in synchronous oscillatory activity when activated by visual stimuli. The frequency of these oscillations was in the range of 40 Hz and differed from the periodic activation induced by the grating, suggesting that the oscillations and their synchronization were due to internal neuronal interactions. Similar findings were shown in parallel by the group of Eckhorn, providing further evidence for the functional role of neural synchronization in feature binding. Since then, numerous studies have replicated these findings and extended them to different modalities such as EEG, providing extensive evidence of the functional role of gamma oscillations in visual perception. Gillis-Laurent and colleagues showed that oscillatory synchronization has an important functional role in odor perception. Perceiving different odors leads to different subsets of neurons firing on different sets of oscillatory cycles. These oscillations can be disrupted by GABA blocker picrotoxin, and the disruption of the oscillatory synchronization leads to impairment of behavioral discrimination of chemically similar odorants in bees and to more similar responses across odors in downstream I-squared lobe neurons. Neural oscillations are also thought to be involved in the sense of time and in somatosensory perception. However, recent findings argue against a clock-like function of cortical gamma oscillations. Motor coordination equals. Oscillations have been commonly reported in the motor system. Virchella and colleagues found a reduction in alpha and beta oscillations in EEG activity when subjects made a movement. Using intracortical recordings, similar changes in oscillatory activity were found in motor cortex when the monkeys performed motor acts that required significant attention. In addition, oscillations at spinal level become synchronized to beta oscillations in the motor cortex during constant muscle activation, as determined by MEG-EEG-EMG coherence. Recently it was found that cortical oscillations propagate as traveling waves across the surface of the motor cortex along dominant spatial axes characteristic of the local circuitry of the motor cortex. Oscillatory rhythms at 10 Hz have been recorded in a brain area called the inferior olive, which is associated with the cerebellum. These oscillations are also observed in motor output of physiological tremor and when performing slow finger movements. These findings may indicate that the human brain controls continuous movements intermittently. In support, it was shown that these movement discontinuities are directly correlated to oscillatory activity in a cerebello-thalamo-cortical loop, which may represent a neural mechanism for the intermittent motor control. Memory. Neural oscillations, in particular theta activity, are extensively linked to memory function. Theta rhythms are very strong in rodent hippocampus and entorhinal cortex during learning and memory retrieval, and they are believed to be vital to the induction of long-term potentiation, a potential cellular mechanism for learning and memory. Coupling between theta and gamma activity is thought to be vital for memory functions, including episodic memory. Tight coordination of single neuron spikes with local theta oscillations is linked to successful memory formation in humans, as more stereotyped spiking predicts better memory. Sleep and consciousness. Sleep is a naturally recurring state characterized by reduced or absent consciousness and proceeds in cycles of rapid eye movement and non-rapid eye movement sleep. The normal order of sleep stages is N1, N2, N3, N2 or REM. Sleep stages are characterized by spectral content of EEG, for instance, stage N1 refers to the transition of the brain from alpha waves to theta waves, whereas stage N3 is characterized by the presence of delta waves. Development. Neural oscillations may play a role in neural development. For example, retinal waves are thought to have properties that define early connectivity of circuits and synapses between cells in the retina. Pathology. Specific types of neural oscillations may also appear in pathological situations, such as Parkinson's disease or epilepsy. Interestingly, these pathological oscillations often consist of an aberrant version of a normal oscillation. For example, one of the best-known types is the spike and wave oscillation, which is typical of generalized or absence epileptic seizures, and which resembles normal sleep spindle oscillations. Tremor. A tremor is an involuntary, somewhat rhythmic, muscle contraction and relaxation involving to and fro movements of one or more body parts. It is the most common of all involuntary movements and can affect the hands, arms, eyes, face, head, vocal cords, trunk, and legs. Most tremors occur in the hands. In some people, tremor is a symptom of another neurological disorder. Many different forms of tremor have been identified, such as essential tremor or Parkinsonian tremor. It is argued that tremors are likely to be multifactorial in origin, with contributions from neural oscillations in the central nervous systems, but also from peripheral mechanisms such as reflex loop resonances. Epilepsy. Epilepsy is a common chronic neurological disorder characterized by seizures. These seizures are transient signs and/or symptoms of abnormal, excessive or hypersynchronous neuronal activity in the brain. Applications. Clinical endpoints. Neural oscillations are sensitive to several drugs influencing brain activity. Accordingly, biomarkers based on neural oscillations are emerging as secondary endpoints in clinical trials and in quantifying effects in preclinical studies. Theses biomarkers are often named EEG biomarkers, or neurophysiological biomarkers, and are quantified using quantitative electroencephalography. EEG biomarkers can be extracted from the EEG using the open-source Neurophysiological Biomarker Toolbox. Brain-Euro-computer interface. Neural oscillation has been applied as a control signal in various brain-Euro-computer interfaces. For example, a non-invasive BCI interface can be created by placing electrodes on the scalp and then measuring the weak electric signals. Although individual neuron activities cannot be recovered through non-invasive BCI because the skull damps and blurs the electromagnetic signals, oscillatory activity can still be reliably detected. In particular, some forms of BCI allow users to control a device by measuring the amplitude of oscillatory activity in specific frequency bands, including mu and beta rhythms. Examples: a non-inclusive list of types of oscillatory activity found in the central nervous system: delta wave, theta wave, alpha wave, mu wave, beta wave, gamma wave, sleep spindle, thalamocortical oscillations, subthreshold membrane potential oscillations, bursting, cardiac cycle, epileptic seizure, mathematical modeling of electrophysiological activity in epilepsy, sharp wave ripple complexes. See also, computational neuroscience, systems neuroscience, neurocybernetics, cybernetics, dynamical systems theory, electroencephalography, magnetoencephalography. References. Further reading: Buzzer Key, Gyao Paragraph Gy. Rhythms of the Brain. Oxford University Press. ISBN 978-0-19-530106-9. Freeman, Walter. Mass Action in the Nervous System. Academic Press. ISBN 0124120474. http Sulcus Berkeley Edu MANS www.html. External links: Binding by synchronization, neural field theory, spike and wave oscillations, synchronization, bursting.